Let's talk about karyotypes for a second. We mentioned this the other day when we were looking at your autosomes, which remember are 1 through 22, and your sex chromosomes, which is your 23rd pair down here. But a karyotype in general is simply a display or a picture of an individual's sets of chromosomes. All right. So when we take a look at these, we're usually pulling these out. Somebody's doing this in a lab under a scope, and they're looking at the way that these chromosomes line up in your cells during typically mitosis. All right, and then they're sorting those chromosomes based on size, and either in this case they're based on color, but a lot of times there'll be bands here, the way they stain these chromosomes, so they'll look for the banding patterns that are the same as well. All right, and what they're doing is taking all of your chromosomes in your cells and pairing up the chromosome 1 that mom gave you and the chromosome 1 that dad gave you. So this would be a homologous chromosome. All right, so information here, let's just say it's eye color from mom, that same genetic information from dad for eye colors over here. All right, so it's on the same location or locus on that homologous chromosome. All right, so what's the purpose to doing karyotypes? All right, well, one of the ways they can do that is to determine the sex of your fetus. Remember, a fetus is a baby growing in the womb. All right, so they're looking really for this here. Now, our technology, especially with ultrasounds, has become much better. The pictures we get are much clearer than they used to be. So that use has sort of fallen away. It's not completely gone because we can only see external sex structures on a fetus at a certain week. So if they want to know beforehand, this is an option, although it's much more invasive. But more often, they're using it for this purpose here. All right, they're looking for chromosomal disorders, which is what we'll get to in just a second. How do these disorders show up? All right, but one thing they might be looking for are extra chromosomes. All right, so something like Down syndrome, which we'll talk about as trisomy 21, tri means 3, so there's actually 3 sets or 3 chromosome 21s instead of the 2 that would be normal. All right, that then leads to some developmental and other physical characteristics that we see of a typical Down syndrome patient. All right, they may also find extra sex chromosomes, so 2 X's and a Y, or only 1 X, something like that. Those also have specific genetic disorders behind them. All right, so that's one thing they may be looking for. So here is kind of what I was talking about. So they'll take a blood culture. If they're doing this from a baby, then that blood culture will have to be taken from mom's stomach, from the baby, so it does become a little bit more of an invasive procedure, so they try not to do it unless it's absolutely necessary. They'll send it through a centrifuge so that they get all your kind of red blood cells and other things down here, and you get more of your plasma, which is the liquid stuff of your blood up here. All right, they'll make a solution out of it. You don't have to know all of this, but they'll ultimately get it onto a slide that they can look at, that they can stain, so that they can see your chromosomes that look like this. All right, and here's the banding pattern I was talking about before. All right, so here this woman is doing this in the lab. Here it's under the scope, and she's got a nice monitor hooked up to her eyepieces so she doesn't have to look through the scope itself. And she's going through and she's sorting out those chromosomes, those homologous pairs, to make something that looks like this. All right, so here's 1 down to 22, your autosomes, and here's the sex chromosome here. Okay, so that's the general idea. All right. So simply be familiar with what a karyotype is and really what its purpose is, which is what we were talking about back here. Okay.